Hypersensitivities are often generally described as allergies, but it's a little bit more than that. The type 1 hypersensitivities were discussed in a little bit more detail in the IgE section and when discussing the function of basophils and eosinophils. So we've already discussed those previous slides or previous sections. What remains are brief descriptions of the other types of hypersensitivities, types 2, 3, and 4. Examples of type 2 hypersensitivities are transfusion reactions and hemolytic disease of the newborn. These types of hypersensitivities involve IgG and complement. Complement is this group of proteins that react or interact either directly or indirectly with antibody that results in cell lysis. The antibody complexes form on the surface of cells, drilling holes into the cytoplasmic membrane, resulting in the rupture or lysis of the cells. Just so happens the cells that we're describing are red blood cells. If you're transfused with the wrong type of blood types come in various forms. Generally we think of A, B, O, AB blood types. And you must really be transfused with our type-specific blood. In addition to the ABO blood group, there are also many other types of red blood cell antigens or blood groups. RH is another. We have RH positive and RH negative antigens. In fact, it's only RH positive antigens that can be found on red blood cells. The absence of RH antigens is what we call the RH negative blood group. The presence of the RH antigen means you are RH positive. And for individuals who are RH negative must never receive or get transfused with RH positive red blood cells. This is what happens in babies who are RH positive whose mothers are RH negative during pregnancy. The immune response to the RH positive antigen is very powerful. It elicits a fairly sort of serious type of hemolytic reaction. In other words, the IgG and complement work together to attack the RH positive cells and result in the lysis of those RH positive cells. The IgG is produced by the mother. The RH negative mother produces IgG, reacting to the RH positive cells of the baby. During pregnancy, there's always a small amount of maternal fetal bleed. So if you take a blood sample from a mother during pregnancy, especially a later trimester, you can find fetal red blood cells circulating in that maternal circulation. Similar things happen if you are artificially transfused with the wrong type of blood. Again, your body has antibodies that will react to those red blood cell antigens and destroy those red blood cells, resulting in cell lysis. Maybe not such a severe problem as with babies, but in both cases, either transfusion reaction or hemolytic disease of the newborn, where your red blood cells are being attacked by IgG and complement, can in fact be lethal in some cases. So we do various types of treatments now, especially with moms who are RH negative, to prevent any unwanted immune reaction of the mother to any potentially RH positive baby cells. For transfusions, we just make certain that we transfuse type specific or the unique type of blood that you need that matches the RH and the ABO configuration of your red blood cells. Type 3 hypersensitivities involve IgG and phagocytes and the formation of immune complexes. Immune complexes are that combination of antibody and antigen that accidentally gets stuck on cells lining various vessels, blood vessels, vessels in the kidney, like the glomerulus, and those immune complexes then become attacked or wind up as targets for phagocytes. It's quite a complex interaction. The illustration tries to show you the formation of immune complexes, essentially deposited antibody antigen on the inner lining of a vessel of some type. Blood vessels are a good example. And then the phagocyte comes along and tries to pull this complex of material from the inner lumen of the cells and essentially, unfortunately, result in all kinds of damage to some of those normal tissues. The type 3 hypersensitivity takes about 6 to 48 hours to develop. So these are a little bit slower in comparison to, let's say, a type 1 hypersensitivity. It's very fast. Type 1s might occur within 15 minutes. Type 3s, again, maybe a couple of days. Serum sickness, where a person has a delayed reaction to maybe a particular antibiotic. The kinds of type 3 hypersensitivities might be called Arthus reactions, cheese handler's disease, pigeon fancier's disease, many different types of antigens that often wind up being inhaled, so they wind up in the alveoli in our lungs, or even tiny little bits and pieces might wind up in blood vessels or lymphatic vessels. These all can trigger, or unfortunately, get complex with IgG, stuck to cell surfaces, and then become targets for the unwanted attack by phagocytes. So type 3 hypersensitivities, again, another category of antigen-antibody interaction that results in even more destruction, unfortunately. Type 4 hypersensitivities are T-cell mediated. They take up to 24 to 72 hours, so these are the slowest to develop. And in fact, often are found when we have reactions to things like poison ivy, poison oak, or poison sumac. These are often contact dermatitis sensitivities if you're allergic to various types of metals, or if you've ever heard of graft versus host disease. If someone is transplanted with various types of body organs, the old-fashioned graft rejection is a slightly different type of reaction, but this is actually where the graft itself triggers a T-cell response that's unwanted. For poison ivy, graft versus host disease, where we have maybe a bone marrow transplant that's unfortunately causing problems for the recipient, or even contact dermatitis. These all essentially involve APCs that process the antigen, present it to a sensitized CD4 positive Th1, releasing interleukins and various other cytokines that further activate antigen-presenting cells that start to attack these various antigens wherever they're located. Often they're on our skin. So we have poison ivy, metal sensitivities, or metal allergies. Next thing you know, we see symptoms of things like eczema, swelling, even that vesicle fluid accumulation that we often associate with the blistering from various types of allergens. For graft versus host disease, bone marrow transplant, that's why we have to destroy the bone marrow and essentially the whole immune system of the recipient, because the immune cells in the bone marrow can actually attack the recipient and unfortunately cause damage to the recipient before even the benefits of that bone marrow can be taken advantage of. So type 4 hypersensitivity is T-cell mediated, and again, the slowest to develop in our time scale of hypersensitivities. In autoimmunities, our immune system essentially erroneously attacks our own tissues, our own cells. Autoimmune. Self. Attacking oneself. We have various types of autoimmunities. Some of them are organ specific. Some of them are what we call systemic. For the systemic autoimmunities, systemic lupus erythematosus is a good example. Occurring when we produce antibodies in the form of IgG antibodies that start to attack our own DNA. These are called anti-DNA autoantibodies. We can detect the presence of these autoimmunity antibodies by various types of immunofluorescent tests. We can actually take samples of blood from the patient, separate the serum antibodies, and localize and find these IgG autoantibodies. We also see symptoms on the patient. In fact, that's how we initially get the preliminary diagnosis of SL, or systemic lupus erythematosus. Oftentimes, patients will display a mask or changes in patterns of pigmentation on their face. It's said to mimic the face of a wolf. That's where we get the wolf name in the disease. Lupus. Lupus is Latin for wolf. So this wolf-like mask, along with various other types of symptoms, often joint pain. In fact, autoimmune diseases are often the diseases that we have to rule out to figure out what the patient does or doesn't have. Autoimmune diseases can be very difficult to pin down or determine, depending on the type of autoimmunity. It results in the formation of immune complexes within blood vessels, within body tissues, depending upon the target. In the case of anti-DNA antibodies, the DNA can be found in any cell in the body. So unfortunately, those immune complexes can form in all kinds of places, within blood vessels, within body tissues. So this is essentially an interesting combination where you're seeing essentially a type 3 hypersensitivity. And unfortunately, the IgG involved in this type 3 hypersensitivity are antibodies directed toward our own antigens. Another example of a systemic autoimmunity is rheumatoid arthritis. With rheumatoid arthritis, the immune complexes form in and around joints within the body. If not reversed, rheumatoid arthritis can be severely disfiguring, resulting in patients not being able to walk or use their hands. The immune response, again, is attacking tissues and cells within our own bodies. So these antibodies, these antibodies that are produced in our bodies, are actually directed in this example to both other antibodies and our own tissues. What a mess. So unfortunately, rheumatoid arthritis and systemic lupus erythematosus, reasonably common types of autoimmunities, especially seen as we age, and our bodies lose the ability to distinguish between self and non-self. Examples of organ-specific autoimmune diseases are conditions like Graves' disease, insulin-dependent diabetes mellitus, In those examples of Graves' disease, the antibodies that are produced are directed toward cells within the thyroid. With IBDM, we have cells that are being destroyed in the pancreas. In these examples, this is more like a type 4 hypersensitivity, with the IBD also involving T-cell destruction of the pancreas. So this combination of antibody-mediated autoimmunity in the case of Graves' disease and T-cell cytotoxicity in the case of IBDM both lead to different types of organ-specific autoimmunity. Immunodeficiencies mean either your T cells and or your B cells are not working properly. With a primary immunodeficiency, this means you were born with either T and or B cell insufficiency. If both are not working, or we have no T cell or B cell function, that's described as severe combined immunodeficiency. And the old story of the boy in the bubble, where an infant was born who had no functional T or B cell immunity, placed in isolation in order to survive, is in fact a condition that does occur. It's fairly rare, but primary severe combined immunodeficiency can occur. More than likely, primary immunodeficiencies occur when it's just one or the other of the lymphocytes that are not functioning. T cell or B cell primary immunodeficiencies can also occur. Secondary immunodeficiencies occur when an individual who's born with a fully functional immune system has something happen to destroy either T and or B cell function. The something that can occur to destroy T or B cell function could be exposure to radiation, chemicals, or different types of environmental factors that might result in the destruction or the impairment of T and or B cell function. A more common example, unfortunately, is the retrovirus HIV. Human immunodeficiency virus infects CD4 positive T lymphocytes. Unfortunately, by gaining entry into the CD4 positive cells, it also impairs the function of those CD4 positive cells, essentially resulting in an acquired immunodeficiency or secondary immunodeficiency due to a virus. So again, different types of infections like HIV, exposure to chemicals, radiation, various factors can result in the impairment of both T and or B cell function, leading to secondary immunodeficiencies. Why do different people seem to be predisposed to various different types of diseases? But sometimes we see diseases run, we say, in families. Well, it all stems back to our self-markers, our MHC. For certain types of diseases, and this is a process still sort of being studied, there seem to be correlations between your particular MHC class 1 and 2 markers and certain types of diseases, often initially triggered by some type of infection with a microorganism, either bacterial or viral, or even parasite. We can determine what is your human leukocyte antigen type, or your HLA, by taking white blood cells from lymphocytes from your body and determining the various different categories of class 1 and class 2 markers. If we find that you fall into a particular category, let's say you have an HLA B27, then there's a particular sort of percentage or likelihood in which you might develop certain types of diseases simply because of this association with this self-marker. Again, scientists have been working for years to try to determine the why and the how that this correlation occurs. And more work is elucidating or determining just how these factors take effect or interact. But what diseases are you predisposed to? It all comes back to your own self-markers and your own immune response.